And I'm officially saying hello and welcome everybody. My name is Mary Abbott and I'm with the Friends of the Sierra Nevada Field Campus. And I'll tell you about the field campus in a second, but the Friends group is an independent nonprofit that was formed to support this program. And the Sierra Nevada Field Campus is a program of San Francisco State University through the College of Science and Engineering. And it's a small, I think it's 10 or 11 acres on the North Yuba River. Um, and it's a place where you can take classes in nature, science, art. And there's some kind of unusual things like fly fishing, and we have a summer camp for adults. Brian's class, the Fungi of the Northern Sierra, is a 4 credit class. So we have about 6 classes that you can get college credit for. You don't have to take the credit, but you can. And then the rest are workshops, which don't give you college credit. And if you don't know where the field campus is, if you flew into Reno, you'd have about an hour and a half drive going west to get there. Or you could drive up Highway 80 to Truckee and cross over to the west to Highway 49, come over the Yuba Pass, and you would come to the field campus. Or you can drive up Highway 49 through Downeyville, and about 20 miles past Downeyville, you'd be at the field campus. And the area is it's such a fascinating area because it's where the Sierra is coming into conjunction with the Cascades. And so it creates this very unique environment of many different habitats and biomes. There's right in the area, we have the Gold Lakes Basin, and there are about 25 lakes within walking distance from wherever you might park. So it's a pretty special place. And the field campus has been around for over 50 years. And of course, we haven't had classes last year or this year in person. We've been doing these virtual things. But we're really got our fingers crossed for next summer and a full calendar. Um, so Brian Perry teaches the fungi of the Northern Sierra Nevada, and that's usually the first class of the year. It's early in the year, last week of May usually, and of course you're going to hear all about it. And we're doing a Zoom to get you excited about this incredible class, and also, we've asked people to donate if you can, because we have a fund going to remodel the lab that Brian uses, and it's on the ground floor of our main building, and it's kind of a cold, dark place, especially at the end of May when sometimes it can still be snowing, and there's not really any heat down there. So anyway, if you can donate, I'm going to be putting a lot of links for you in the chat, ways to connect with the Friends of the Sierra Nevada Field Campus, ways to donate, how to reach Brian, a species list of the fungi in the Northern Sierra. And we have Facebook groups, and that's a good place to sign on because you get kind of up to the minute information about the field campus. And I also want to welcome, we've got a couple board members here. Stephen Kulicki, if you want to say hi, he's our Facebook PR guy. And Diane and Bill Wilson are here, but you're hiding behind your cameras are hidden. Diane and Bill Wilson, Bill is the president of the Friends, and they have been going, every single year to your class, Brian? Yeah, many
Yes,
years.
they're
Yeah.
alumni.
Yeah, they knew you when you were just a student, right?
Yes.
Yeah. So, and if I haven't forgotten anything, maybe we can get started. Take it away, Brian.
All right, sounds good.
Okay.
Well, thank you, Mary, for the introduction, and thanks for inviting me. It's always fun to be able to talk about the field campus and about the course up there. And thanks everyone for coming. Hopefully everyone's well into their dinner now, maybe having a glass of wine. Hopefully I can entertain you and not put you to sleep. But yeah, my goal tonight is to really tell you about the field campus, to tell you about the course that I'm lucky enough to teach up there, and really introduce you to fungi if you don't know much about them and you're someone who's interested in learning. And if you are someone who's into fungi, hopefully I can tell you something new about them. And my real goal here is to get you excited to come up and take not just my class, but really any class at the field station, once we're back up and running, hopefully next year, fingers crossed. So with that, I'm going to go ahead and share my screen. I've got a PowerPoint presentation for you all here. All right. So before I get too far into it, if you folks have any questions or anything during the talk, because of the way I'm sharing my screen, I can't see the chat or anything like that, but feel free to type in questions. And Mary, if you see anything and people are having issues or want me to address something, you can just stop me at any time. We can make this as informal as possible.
Yeah, I'll watch the chat for you. And do you want me to save questions to the end, Brian?
Yeah, that'd be
Or
great. I can get through
okay.
this, and then if folks have questions, we can chat afterwards.
Oh, good. Okay, good. Ask away, everybody in the chat.
All right, perfect. All right, so the official name of the class is actually Spring Fungi of the Sierra Nevada. So I've always thought about changing the name to Spring Mushrooms of the Sierra Nevada, because what we're doing during this week up here in the Sierra is we're really looking at the mushrooms, which are the reproductive structures of fungi. So as you probably, or most of fungi are incredibly diverse and they're incredibly variable in their morphologies and their lifestyles and where we find them on the planet and in the environment, wherever you may be. We're really focusing on, for the most part, macroscopic fungi of the Sierra Nevada during this first week. So for a lot of folks, this is their first introduction to fungi. So they're going to learn how they basically go out in the environment and find these things and bring them back to the laboratory and identify them. And then we're also going to teach them to take that to the next level and learn how to use a microscope to get even further in their identification process and to really hone their skills. So it's a week-long course. As Mary said, it's the first course taught at the field station. So we're that first week. And there's usually another class going on while we're there. That might be a birding class. It might be a watercolor class. So there's always variation. The schedule is always changing around a little bit. But because fungi are so dependent upon moisture and temperature, we always teach this course during the very opening week of the season up there. Okay, and so this year, had we taught it, we would have started on Memorial Day and gone from May 31st, up to June 4th or something like that would have been the length of the class. So introduce myself, I'm Dr. Brian Perry. I'm a mycologist by training. So I teach a number of courses at Cal State East Bay. I teach 2 mycology courses there. I teach general biology, I teach evolution, I teach a bunch of phylogenetic analysis courses. So a bunch of different things. Mycology is my first love and really my true love in the sciences. So it's always really special for me to be able to come up to the field campus and offer my course. And to give you a little bit of history behind the course, it was really founded by Dr. Harry Tiers. And Harry Tiers was the original mycologist at San Francisco State University. He taught the very first version of this course back in 1982. And Harry taught the course from 1982 all the way up until the time that he retired in 1989. And it's been kind of a really magical thing where Harry has taught the course and then it's been one of his students. So someone who, viewed Harry as their mentor, excuse me, has taken over the course. So in the photograph you see here, this is Dr. Dennis Desjardins. He looks a little bit different these days. This was back in 1984, but he took over the course after Harry taught and basically taught it all the way up until I took the course over back in 2017, 2018. So just to give folks the credit they're due, Dr. Roy Halling was one of Harry Tiers' graduate students, and he actually married Harry's daughter. And Roy taught the class for 1 year. So the year after Harry retired, Roy came out from New York, taught the course. He was basically working at New York Botanical Garden and retired just about 2 years ago. Another one of Dr. Tiers' students, Dr. Andrew Methvin, came out and taught the course in 1992. He was a professor at Eastern Illinois University for a number of years and just also retired just a few years ago. And then Dr. Dennis Desjardins, who's at San Francisco State University, also one of Harry Tiers' students, and actually came back and replaced Harry as the mycologist in the biology department at San Francisco State. And so Dennis taught the course once in 1991. That was right when he was in the middle of finishing up his PhD. Then he came back after he got his PhD, took Harry's job, and basically taught the course up here from 1993 all the way until 2017. And then I had the luxury of taking it over. So just to keep sort of the tradition going, I was one of Dennis's students. So I did my master's degree work at San Francisco State with Dennis. I came up every single year I was a student there. I came up, I helped Dennis teach the class as a TA in many years, or I just came up and worked on my own research and project. But anything I could do to get up to the field campus, I took advantage of. So Dennis was my mentor, and then he basically handed off the course to me. So maybe someday, hopefully 20 years from now, whatever it may be, I'll hand off the course to one of my students. Hopefully they'll have a new lab by then. That's the goal, right? So June 2019 was the last time we offered this course, and that was the 35th anniversary of the class being taught. There was a handful of years where no course was offered, either because there was no one available to teach it, or we had 1 or 2 years where the drought conditions were so bad that we actually canceled the class because we're afraid there wouldn't be any mushrooms for the students up there. So since the course was founded in 1982, there was 3 years where we didn't teach it. And then of course we've got 1920 and 1921 where we didn't offer it either. So we've got this 5 years where we haven't offered the course up there, but otherwise the course has been going strong. Next time we teach it, it'll be the 36th anniversary of the course being offered. Now I want you to look at this photograph. Look, it's a nice, beautiful sunny morning. People are wearing flannels or maybe a sweatshirt. It doesn't look too cold. Everyone's pretty happy. That's, the kind of conditions that we really hope for up there. But as Mary hinted at, that's not always the case. So because we are up in the Sierra Nevada, usually during that last week of May or first week of June, the weather conditions can be incredibly variable. So this is a picture from 2012. If you look closely at the photograph, you'll see some of you that are at the talk tonight, you'll see your own faces in there, so welcome back. But you'll see that most of the folks in the photograph are holding mushrooms, but there's a bunch of snow on the ground. So this was a week where we all got up there, the temperatures were beautiful, it was in the 70s, it was nice and moist, the conditions were ideal. We'd been out collecting mushrooms for about 2 days, and then suddenly a big storm rolled in and it dumped about 2 feet of snow overnight. We had lots of people's tents were collapsing. It was people were sleeping in the dining hall and the director's cabin and basically trying to find some place to keep warm after their tents had collapsed, or people that didn't have warm enough sleeping bags to sleep in the snow. But we made do with it. Okay. And then about a day later, all that snow melted and the mushrooms were still there. The mushrooms came out. We continued the class. We had a great time. I got to give the 2012 class kudos. Everyone, kept a really positive attitude. They stuck it out. The mushrooms were buried under 2 feet of snow. We basically had to wait for them to melt. But as I'll talk about in a few minutes, what we do in situations like this is we just drive down the western slope of the Sierra Nevada till we get out of the snow, and we'll collect in environments where there's not 2 feet of snow on the ground bearing all the mushrooms. Okay. So to give you an idea of what the way the class runs, just so for those of you that I hope will, I know many of you in the audience have signed up to take the class before and it's been canceled these last 2 years. So I really hope you'll sign up next year and come up. To give you an idea of the way the class works for those of you that are considering it, it starts on Sunday night and it goes until Friday morning. And every day after breakfast, we're going to have a lecture. And that lecture is going to include the basics of fungal biology. We're going to talk about macroscopic features of fungi. We talk about microscopic features of fungi. We talk about fungal ecology. And then we also talk about all the different fungi that we know from the Sierra Nevada. So we'll go through all the different groups of macro fungi that we find up in the Sierra Nevada in this area during spring. That usually lasts for about 2 hours or so. Then I want to wake everyone up from their slumber, give them another cup of coffee. We head out in the field about 10:45. And so we'll do field trips to different habitats in different localities each day. So we have 4 days worth of field trips where we're going to go out to different spots that are all within, maybe a 20 to 30-minute drive from the field campus. Really depends on the environmental conditions and how far we need to go. So we'll spend most of the day in the field. We'll have our lunch in the field, and then we come back around 3 o'clock. And that's when we work in the laboratory. And I'll show you some photographs of the laboratory in just a minute. But we work in the lab from 3 to 6. So during that period, the TA and I will teach you how to use things like dichotomous keys. So the first thing we'll do is we'll teach you all the different portions of a mushroom. So if you're holding a mushroom in your hand, we'll tell you where the stipe is, where the lamellae are, where the pileus is, all the different features and terminology that you need to learn. We'll go over that in the laboratory together as a group. And then over the years, we have written a really lovely field guide, if I can get that to come into focus, if I put it by my face, to the spring fungi of the Sierra Nevada. And this field guide uses both macroscopic and microscopic features. So throughout the week, you'll be using this book, this field guide that we've written to identify all the mushrooms that you find when we're out on our field trips. Okay, and we do that until about 6 o'clock. There may or may not be a happy hour. I won't say more about that. Then we have dinner, a lovely, wonderful meal prepared by the staff at the field campus. And then in the evenings, we have these optional presentations. So one of the really special things about the field campus and the fungi course is that people that come up there and take the class once want to come back every single year and participate. So we have almost as many alumni that you like to come up as we do students in some years. Many of those alumni are professionals, or they're folks that have a specific knowledge or a specific skill set that has to do with fungi. And so we'll have these evening presentations, and these might range on me giving you folks a talk about the work I do in the South Pacific, documenting fungal diversity, or it might be Dr. Andrew Wilson from the Denver Botanic Garden talking about the work that he's doing in Colorado, documenting fungal diversity there. Or it could be something like having Allison Pollock, who's here tonight. When she becomes an alumnus of the class, we could have her come up and give us a talk on the amazing macro photography that she does, right? We can see lots of her amazing slides. It would be really, really cool. So we do these evening presentations. They're totally optional. If you want to come and join us and, have a glass of wine while you listen, that's totally preferred. But if you want to go back to your tent and sleep, you want to go lay under the stars and do something else, that's totally okay. They're 100% optional. So let me give you an idea of what it's like to go out in the field. So every morning we're going to jump in cars, we'll carpool out to our usual field sites. And these are areas where we know that, we can usually count on finding a good diversity of fungi. We also know that they're pretty accessible to a wide range of folks. So if you're not someone who's totally mobile and can't hike up the side of the hill, we'll go to areas where you can easily walk around and collect mushrooms. And this is going to include things like our pine fir forest. So we'll go to some really nice, pristine forest sites up around the Yuba Pass area. We also go to disturbed habitats. So campgrounds are a really great place to collect fungi. Not only are campgrounds nice and flat and open, which makes walking around and seeing mushrooms easy, there's a lot of fungi that will actually fruit. In other words, they'll reproduce and make their fruiting structures, their mushrooms, in these compacted soils that we find in disturbed habitats. So if you're a morel hunter, you know that going into campgrounds is a great spot to find morels where people either drive down the roads or where there's trails or, high foot traffic around campsite tables, all those sorts of things. This can be really productive areas. The other thing that occurs in disturbed habitats like campgrounds are fire pits. And because we're usually up in the area so early in the season, we will walk around and actually look in the fire pit rings. And there are fungi that have evolved to reproduce and take advantage of that sterilized environment that a fire pit creates. So in those areas, you're going to have an influx of nutrients into the soil from the burning wood that's in there, but also that soil gets sterilized. So it sort of will kill off a lot of the fungi so that these sort of early colonizing species will come back into that space. And so we'll find very unique fungi growing in those fire pits, okay? So if you ever see me walking around looking in fire pits, now you'll know why. We're going to go collect in meadows. So we have a lot of beautiful meadows up there. And before they dry out too much, there can be a lot of really interesting grassland species that will come up in those meadows. And then of course, snowbanks. Probably the most unique habitat, or I'll say feature, of collecting mushrooms in the Sierra Nevada in the spring is that they're really dependent upon the moisture produced by the snowpack melting and seeping into the soil. So we have a whole group of fungi that we only find in association with melting snowbanks. They need to be close enough to that snowbank so that there's a significant amount of moisture in the soil. And in many cases, we'll even see those mushrooms just pushing right through the edge of the snowbank. They'll essentially melt their way through it using their heat that they generate from their metabolism. So wandering around snowbanks, and what you'll see is all around the edge of those snowbanks, you'll find these incredibly interesting fungi. And I'll show you some pictures of mushrooms in just a few minutes, I promise. Okay. And also seepage areas. So, looking at this, you see some alders and you've got some sedges and things growing out here and lots of grasses. And this is a really wet area. If you were to walk out into this photograph, your feet would be wet. You'd sink up to up above your ankles and moisture and water. And these are these seeps. And we find these essentially where creeks will come down and sort of spread out into a flattened area. The water will, form these little rivulets and stuff, but it really seeps throughout the soil and creates these really moist, wet environments. And you have to get to there at just the right time. There can't be too much water that's moving because the fungi won't reproduce in that environment. And once this thing dries out, then the fungi won't reproduce at all. So you have to get there at just the right time. And what you'll see is that when you go out into these seeps, there's a handful of species that we get in the Sierra Nevada in the spring, only in these habitats. And they're these incredibly beautiful, really interesting species. So this is the one from my title slide. This is two species here. This is Mitrula elegans, which is the little orange one, which you can see here. This is known as the matchstick fungus, or swamp beacons is what they're oftentimes called. And it's not a mushroom in the sense that it has gills, like your grocery store mushroom. This is actually a member of the phylum Ascomycota, which includes things like morels and cup fungi, if you know what those are. And this is basically a little stalk. Sorry, my mouse isn't working perfectly here. And at the tip of that stalk, this little orange match head, that's where this fungus is reproducing. So that's where this fungus is producing the spores, which are going to get forcefully ejected out of the cells that cover that orange surface. And those spores are going to land in the water. Some will get carried away by wind, but most of them are going to land in the water. And then that water is going to basically disperse them for the fungus. These spores are going to attach themselves to pieces of rotting plant matter. They'll germinate and they'll grow. And they'll use that rotting plant matter as a food source. And then they'll spend the rest of the season there. They'll overwinter there. In the following spring, when the water comes back and it's at just the right level and just the right temperature, they'll reproduce. Okay. Same thing goes for Cudoniella clavis that you see here. This one's a little bit different. This one has more of a turbinate, sort of creamy pink colored head to it, but it's doing exactly the same thing. Now, to give you an idea of scale on these, hopefully you can all see those little tiny yellow dots on top of those. Those are pine pollen grains. So this gives you an idea that these are quite small fungi, maybe an inch tall and a few millimeters in diameter. So pretty small. In the same habitat, we find, this is one of my favorites. This is Sclerotinia sulcata. And this is one that grows on sedges. So it grows on a specific species of sedge, which grows in these seepage areas throughout the Sierra Nevada and also up into the Cascades. Now, we had never seen this fungus in the Sierra Nevada in the 35 years that we've been doing this course up there. We hadn't seen this until about 3 years ago or 4 years ago. We went out to one of our usual meadows called Bear Trap Meadows, where we collect these wonderful seepage area fungi, and this fungus was everywhere. And it turns out what this does is it's kind of a parasite on these sedges. And so the spores, what you're looking at here, this cup-like structure inside that little cup, that tissue or all the cells lining that cup, are where the spores are produced. And when they're mature, they're going to get forcibly shot out of those cells, and they're going to get shot out of that little cup, and then they're going to get dispersed and they're going to land on the surrounding plants. And of course, they're going to land in the water. When they come into contact with a specific species of sedge, they're going to germinate, and those spores are going to basically penetrate into the cells in that sedge, and they're going to grow all the way down towards the base of that sedge that's underwater. And they're going to essentially use that as a food resource as that sedge dies at the end of the season. But then they form these really tough, fibrous, thick structures that we call sclerotia. And these sclerotia are kind of like, hard to explain until you really see one, but it's almost like this carbonaceous hunk of tissue. And it's evolved to really just overwinter. So this thing is going to sit out in the environment. It's going to get covered with snow for the entire winter. Once the snow melts and the waters come back, that sclerotium is going to germinate and grow, and it's going to give rise to these little cup fungi here, which is what we find in the spring. So that is the reproductive structure of that fungus. And I should have said it earlier, if any of you, fungi are brand new to you, a good analogy to think of is apples and an apple tree, right? So a mushroom is basically like an apple is to an apple tree. If you think about it, the apple that we pick off a tree is just the reproductive basically unit of that plant, right? We know that apple contains seeds. Those seeds are the reproductive propagules of that apple tree. The rest of that plant is just this giant tree that's out there photosynthesizing and going through flowers, basically waiting to go through reproduction every season. Same thing for a fungus. The real business end of this fungus is this huge network of long filamentous cells that we call mycelium. That's what's penetrating out into the environment and either acting as, a saprotroph, breaking down dead organic matter, or as a parasite or pathogen, or as a mutualistic symbiont. The mushroom that these fungi produce, that is simply produced only so that the mushroom, or so the fungus, can undergo reproduction. Okay, so it's just like an apple on an apple tree. This is just the mushroom on the mycelium. All right, before I get too far, these are sort of the main habitats that we can find within a short driving distance of the field campus. So we're going to go, down the hill from the field campus, then we go all the way up to Yuba Pass, and we'll drive all the way out Weber Lake Road till we can get up to the highest possible elevation there. And then we get some different plant hosts. We'll get hemlock trees instead of our pine and fir trees, things like that. If it's a year where there's still a huge amount of snow on the ground, or it's snowing on us, or it's really cold, oftentimes we'll drive down the hill towards Sierra City, if you're familiar with the area, and we get into a habitat where there's oak trees. Lower elevation, it's usually drier and warmer. So some years we can even go down pretty far in elevation from the field station and collect mushrooms down there. So throughout the week the class is offered, you're going to get to collect basically from about the 5,000 foot elevation all the way up to the 7,000, 7,500 foot elevation. So a huge range and lots of different habitats. You'll learn, how to find mushrooms in those habitats. All right, so what I have here, this is just, it's a video, it's about 5 minutes long. It just really gives you an idea of what it's like to walk around the woods collecting mushrooms. Okay, so I'm gonna let this play. I'm gonna unplug my headphones so you can hear it. And we'll play this. Well, we're in the Sierra Nevada. It's June 3rd. Normally we'd be having our spring fungi of the Sierra Nevada course, but due to the pandemic this year, we had to cancel it. But I came up anyways to check on some of the field sites and decided to take some videos so I could show all you folks what it's like to collect mushrooms in the Sierra Nevada in the spring. So here I am walking along the North Fork of the Yuba River. This is the field station owned by San Francisco State University. And I'm just looking around for mushrooms. And in the spring, when you're searching for mushrooms in the Sierra Nevada, oftentimes you're going to be looking for little lumps, or what are referred to as mushrumps or shrumps in a lot of the field guides. And what's going on here is that the fungus is producing its basidiomes, its reproductive structures that is, a few inches under the soil. And as these mushrooms expand and grow, they're going to push the soil up. So when you're collecting mushrooms up in the spring, oftentimes you're not walking through the woods looking for mushrooms just sticking out of the ground, but you're actually looking for shrumps or mushrooms. And walking through the woods now, I can see many of them. And I'll stop at a few up here. And hopefully you can see them on the camera. It's very difficult for me to tell. But as I'm approaching this right now, you can see here's a mushroom that has begun to push up and poke through the soil. And so what I would do in this situation is I would come and I would begin to dig gently around the mushroom. And so what you see is we have the cap or pileus of the mushroom sticking up out of the soil. And I'm lucky because this is one of our spring porcini. This is Boletus rex veris. This is very common up in the Sierra Nevada in the spring, in certain years, if you get up here at the right time and there's been a good amount of snow melt and rain and the temperatures are just right. And so what I do now is I take my collecting knife, I'm going to dig underneath and just sort of pop that mushroom out. And so what you see here is we have a nice little porcini or spring king or Boletus rex veris button. And it's a beaut. It's rock hard. This one will be good to eat if you're so inclined. And as you can see, this has pores on the underside instead of gills, okay? And these things have pushed their way up through the soil, okay? Now, looking around here, so when you find one, you always want to look and see if you can find some more. So looking around the area here, I spy over here a few more shrumps. So looking at the ground right here, I can begin to dig gently, and sure enough, there's two very small porcini buttons there. And then when I look over here to my right, I see even larger shrumps. So looking at these ones, let's see what we've got. Oh my goodness, look at that. Here we have a beautiful young cluster of Boletus rex veris just pushing their way up through the soil.
I'm not going to collect these because these still have yet to really grow and they haven't had a chance to disperse their spores yet. So I'm going to leave these here. Because I don't want them to get dried out by the sun, I'm going to kind of cover them back up with some of the soil here, just so that they don't get exposed and dry out too much. They'll continue to expand and grow and push up through the soil and begin to disperse their spores. And then over to the other side here, you see there's another shrub. And if I start pulling this one back, we're going to see something similar, right? This is undoubtedly the same individual probably producing lots and lots of basidiomes or mushrooms. And again, these are very young, so I'm going to leave them in place, leave them be. And so I'm going to look around and see if I can see some more shrubs. There's a few right here, again, young basidiomes that I will leave in place. There's a shrub right there you can probably see. Looking around, here's another shrub. Let's see what we have here. Again, a nice cluster. And you'll notice as I'm doing this, we're getting all sorts of insects that are flying out. And if you're someone who collects foliates a lot, you know that a lot of times when you cut them open, you'll see little maggot tunnels in them. So what's going on here is that these flies that you see buzzing out of here as I push the dirt back, those flies are actually laying their eggs on the pileus of the mushroom there. And those larvae, the maggots, are going to essentially crawl through into that mushroom and use it as a food source. Okay, so we're going to cover those back up, let them be. Looking a further bit over, I see something else coming up here. See what we got here. This is not a porcini. This is going to be a different species. I think I know what this is. I'm just going to pop it up here and have a look. And sure enough, this is going to be one of our, and give it a smell, this is going to be one of our hygrophorus species. And this looks like it might be Hygrophorus purpurascens because it has these little sort of purplish stains right where the gills meet the stipe. And again, this is another fairly common species that we get up here during the springtime after the snow has melted. So we'll just leave that there to disperse its spores. Looking around some more, see if I can find some more shrubs in this area. Here's one right here. I bet this will be more porcini. Yep, so I'm just gonna see just the caps there. You'll notice there's lots of red ants all over the place in there, so I'm gonna leave that one alone. And then here's a nice big one right behind me that I missed. Pushing this one back again, see all those flies coming out of there. Some young buttons, which I'll leave in place to continue to grow and produce their spores. All right, so that's mushroom hunting in the spring in the Sierra Nevada. And again, here's another beautiful little Boletus rexbearis just pushing its way up through the soil.
All right. Well, that makes me wish I was up there right now collecting mushrooms. Okay, so that's hopefully that gives you folks an idea of what it's like. I know many of you have probably been up there collecting mushrooms before. Perhaps you've collected mushrooms along the coast, but we really do see a sort of a difference in the way mushrooms are formed because of the temperatures and the fact that the soils dry out so quickly up there. We do see fungi fruiting a little bit deeper and pushing their way up through the soil. Now, this isn't going to be the case for every mushroom up there, obviously, but it is really common. Okay. So now what I want to do is go through and just show you some of the absolutely gorgeous fungi that we have up in the Sierra Nevada in the springtime, and sort of tell you about some of the more special ones, to me at least. And so for me, the really special thing, as I mentioned before, is going up and collecting around these snowbanks and finding what we call snowbank species or snowbank fungi. Now, these are fungi that are associated with that melting snow. And as you can see from the top two pictures, both of them, whoops, sorry about that. Both of them are really closely associated with that melting snowbank. In fact, Mycena navicula here has melted its way through just the edge of that snowbank. So that fungus, as it's going through its cellular respiration, metabolism basically, it's generating heat just like we do. And that heat is enough for it to melt its way up through the snow. Keeping in mind that, the whole goal of these mushrooms is to produce spores. Spores are the reproductive propagules of this fungus. They're analogous to a seed in a plant to a degree. And these spores are going to get dispersed out into the environment. So these fungi have evolved all these amazing ways to get that spore producing structure up in the air so that things like wind currents can help carry those spores away and disperse them for the fungus. So this is Mycena navicula. This is one that I worked on and actually described as a new species from the Sierra Nevada as part of my master's thesis work with Dr. Desjardins at San Francisco State University. Really common. You will see it up there every year. All you got to do is go and find some snowbanks or where snowbanks have been in the previous few days, and you can find just big clusters of this coming off the soil or woody debris in the soil. One of my other favorites is Donadinia nigrella. This is an ascomycete. So this fungus doesn't have gills. This one does. It's hard to see, but if we flipped it over, you'd see gills on the underside of Mycena here. For Donadinia, this is another cup fungus. So what you get are these black or grayish or smoky black goblets that are growing right on the edge of the snowbank, which can make them almost impossible to see because the soil and the rotting vegetation are super dark, the snow is super bright, and that contrast makes these pretty challenging to find at times, especially if you're walking around in the full sun. But what these are, they're these beautiful goblets that they have this long stalk, and then you get this cup at the very top. And that little cup forms a goblet, and lining the inside of that goblet, this dark, smooth surface that you see here, it's just packed full of cells where the spores are being produced. Those spores are going to get launched out of those cells forcibly through turgor pressure. They're going to get, shot up into the air, and then wind currents are going to come and help disperse them for that fungus. Okay, so that's Donadinia nigrella. Here's Clitosobe glacialis. Again, this is one that we typically find growing right along the edge of the melting snowbanks, hence the glacialis term in the species name. And it's really common. This is one of the most common mushrooms we can find most years, I should say, up in the Sierra Nevada in the spring. And you see it's got this really sort of it's a bloom, is what we say. Basically, the surface of the pileus has these cells that create like a bloom. So when you go and you collect fresh blueberries, they have a bloom on them, and that's kind of wax really. But as you rub it, it disappears. You kind of get the same effect with this. These cells, once people touch them or handle them, or plants brush against them, it basically compresses those cells and that bloom goes away. But these mushrooms start out with this beautiful whitish gray bloom. And then once that goes away, there's sort of this chestnut brown underneath. And then they've got, these gills and then your basic stipe. So Clitosobe glacialis, really common. And then we have things like jelly fungi. So Heterotextus alpinus, this is our most common jelly fungus in the Sierra Nevada in the spring. It looks like orange gumdrops on sticks or logs. So again, you'll find logs that are protruding from melting snowbanks, or they're sitting right next to a melting snowbank. So they're really, really wet and soggy and waterlogged. The whole surface of those sticks or logs or branches can be covered by this jelly fungus. And what's going on here is that if we look at this upper surface right here, that's where all the spores are being produced. The cells that produce them are all embedded in this really gelatinous, sort of gumdroppy texture. And then the spores are produced right at the surface where they get basically blown away, or rain will come and rinse them off the surface there. Okay, so these are jelly fungi. What's pretty cool about jelly fungi, at least this species, is that it has the ability to dry out, when it's in the full sun during the day, and then it will just rehydrate in the morning and produce its spores, and then it will dry out, and it can do that repeatedly. Okay, so they've evolved and they've adapted to do really well in these incredibly arid environments that we have in the high elevation, as long as there's surface moisture around for them. So those are snowbank species. We also have a lot of edible species, and many folks come up and take the class because they want to learn how to identify mushrooms for the table. And that's totally fine. We get a handful of really phenomenal edible species up in the Sierra in the spring and the fall. In the spring, we get the spring kings, what is called Boletus rex varis is the species name. And we also have at least 3 or 4 or more species of morel, or members of the genus Morchella. And both of these are fantastic edibles. And in a good year, so this is what I was collecting in the video I just showed you. That was filmed last year. And last year was a bolete year. We went up there for the same week that the class should have been taught. And it was pretty dry. We hadn't had a lot of rain or anything like that, but we got there just at the perfect time when the boletes were fruiting. So we collected, many, many pounds of this that we shared with friends and family and stuff that we could go and drop it off at their house during the pandemic and give them a little care package. But this is the spring king. It can be incredibly abundant and common certain years while we're up there, and other years we're up there and we don't see it at all. So it's one where you have to be there at just the right time. You've got to be at the right elevation at the right time. The stars and moons have to be aligned perfectly. There has to be just the right amount of soil moisture. The temperature has to be perfect. And then these things will, produce all over the place. And so some years are really great. Some years we see very few of them. This year, 2021, when I was up there, I believe we only found 1 or 2 of these. So it wasn't a great year for them. It was really hot and dry that first week of June, and it wasn't good conditions for the boletes. I don't know if they came up later. I haven't been back up since, but in good years are great. And when we're lucky, the students will go out and collect lots and lots of these and share them with the class. So the kitchen staff at the field station has been phenomenal. They love cooking mushrooms if we bring them to them. And so every year when we go out and collect boletes, if we have enough of them, we'll, give them a huge giant bag or basket full of them, and they'll make us some really fantastic meal that includes these. Or if we have just a small amount, they'll let us take over the kitchen and just cook them so that everyone can have a taste, right? And the same thing with morels. Morels are another, everyone's favorite. Folks go up to collect those. And again, certain years we find tons of them because the environmental conditions are just right. Other years we find very, very few. Okay, so there's other edible species up there to collect and eat, but these are the two most commonly and sought after that we have up there. And then rare species. For me, as a scientist, these are the ones that are the most exciting to me. So we have a number of species up in the Sierra Nevada and really everywhere we go that are really rare. And what I want you to keep in mind is that we've been collecting the same field sites and the same habitats and locations for over 35 years up where the field station is located. Every year we find things we've never seen before, right? So it could be something where we just happen to be there at a slightly different, environmental conditions than we normally are. Or this could be a fungus that's been in the environment the whole time we've been there, but it's just now reproducing because environmental conditions are right. Cantharossa gruberi, this is one that we had never seen until about 10 years ago, maybe 12 years ago now. And we were right down the street from the field campus and we found this thing. This is big, actually the one that's in the photograph here is this big cluster of them fruiting. And this is a really big, hard, robust mushroom. Notice that it's not underground either. It was right on top of the soil where you could see it. And we'd never seen this thing before. We've been collecting that area for at that point in time, over 25 years. Had never seen it. Turns out it's a pretty rare fungus for us. There's probably maybe 10 collections or 10, records of this species known from the Sierra Nevada at this point. It does occur in the Cascades, and I believe it does occur in the Rockies as well, but incredibly rare for us in the Sierra Nevada. Same thing goes with Hygrophorus ceruleus. This is one that we usually find this every year, but when the conditions are good, but not every year. I shouldn't say not every year, but we don't find it in abundance. Okay, so this is one for us that can be pretty darn rare. I've never seen this mushroom fresh. Okay, so this is one that I'll go out and look for every single year. And then another one, Hygrophorus getzii, which is this beautiful pinkish mushroom here. This one grows with melting snow at really high elevations. So what you're looking at here, these are the needles of a hemlock. This one grows with our mountain hemlock. You have to get up in pretty high elevation to get into hemlock stands. We do have one up at Yuba Pass. So every year, assuming the road isn't blocked by snow, we'll drive out to the hemlock patch with the students, and we'll go out, we'll try and find Hygrophorus getzii, just because it's a pretty special rare fungus for us. And then, of course, as I alluded to, we find things that are new, things that are undescribed and new to science. And so there's a number of species that have been described from, from the area where the field class campus occurs and where all the students go out and collect. And there's a number of things that have been collected during that week the class is taught. One of these is Lactarius rubriviridis. I know those of you that know Lactarius probably think I have the picture mislabeled here, but it's not. This is a Lactarius that has basically become a truffle. So it's really it's folded in on itself. Its gills have become all convoluted and it's moved underground. It's become hypogeous, so under earth, hypogeous underground. If you find this thing and you cut it open, there's all these little chambers. Those chambers are where all the spores are being produced. Those spores, because this thing lives underground, will just get dumped right into the soil as this mushroom rots. And that's going to let them percolate through the soil where they can germinate and grow and then come into contact with the roots of trees, pine trees and fir trees, which they form mutualistic symbiotic associations with that we call mycorrhizae. Okay, so this is one that Dr. Desjardins found, described it as a Lactarius based on both morphological features as well as DNA sequence data. And it made people pretty unhappy. People thought that he should put it into one of these older genera that all these truffle-like things are put in. he was following a sort of modern approach. And so this is a Lactarius. Nowadays, everyone's doing this. Okay, so if you find something, you look at his DNA and say, well, this is a truffle-like thing that falls out right in the middle of this normal guild mushroom genus. We just describe it as a member of that genus, which is the most proper thing to do. Stribulus diminutivis, again, this is another one. This is one that I collected and found during my master's thesis work. So for my master's thesis, I worked on the genus Mycena from California. And this little tiny mushroom that you're seeing here, those little dots that you see in the background and all over down at the base, those are pine pollen grains. This is a fungus that grows only on the bracts or the scales that make up the cones of primarily sugar pine, which are these really big cones that we get up in the Sierra, and a few other pine species in the Sierra. If you get those cones and they're pretty waterlogged and wet, if you start peeling back the little scales, the little woody scales that make up that cone, you'll see these little tiny mushrooms growing on them. That's Stribulus diminutivis. And so this is one that I discovered during my master's thesis work, and Dr. Desjardins described it as the species. And then the final one, this is Chetotirsia vernalis. This is a ascomycete, so it's a cup fungus. And what you're looking at is they're just sort of these white or cream colored cups that have this little fringe of hair along the margin and a little bit of hair on the outside. And then you get this big cream, smooth white surface. That's where the spores are being produced. And this is one where the spores are going to get forcibly ejected up into the air again, and air currents are going to carry them away. And this is one that we discovered back when I was working on my PhD. We decided we wanted to honor Dr. Harry Tiers, since he was the founder of this class. He was also the head of the Harry Deer Tiers Herbarium at San Francisco State University. We wanted to honor all of his contributions to mycology, so we wanted to name not just the species, but the whole genus after him. So it took us a while to come up with it, but we came up with Chetotirsia. So Cheto means hair or hairs, and Tirsia is just in honor of Tiers. So this is really the Harry Tiers genus, and vernalis is in reference to this thing fruiting in the spring. Okay, so that's Chetotirsia vernalis. We see it every year now. It was probably there all along, but no one took the time to really look at it and study it until I did so during my PhD work. All right, and finally, we get back to the lab. So we spend all day out in the woods, walking around, having a good old time, collecting mushrooms, having our lunch, and then we come back to the lab. And this is the lab that Mary was talking about, that the Friends of the Sierra Nevada Field Camp is trying to raise funds to renovate and improve. And when I look at this, the pictures that you see on the right-hand side of the screen, there's pictures of people in there that are now at least 20 years older. Okay, so these are old pictures from when I was a student, but the lab looks exactly the same. So the lab has not changed much. If you look over here, you'll see on this wall here, you'll see some construction. That's because a big snowbank actually slid into the back of the cabin and undermined and pushed that wall in a number of years ago, so it had to be repaired. So the lab has been, it's been used every year for 50 years now, and it's showing its age. And so the field Friends of the Sierra Nevada Field Campus have been so amazing and gracious that they've been raising funds that we're hoping we can eventually use to renovate this lab space and kind of make it a little bit more bright. So maybe some more windows, maybe some heat, because it does get incredibly cold down there if we're certain years, and some, just better light and everything else for teaching down there. So that's the lab. I can also, one thing we have upgraded is the microscopes. We do have different microscopes now, but all these old tables that you see are still the same tables that we're using. So the Friends have been fantastic in providing us with new equipment down there. Now, this is what it looks like when we get back from the field. So every student will be assigned to a spot. Every spot has a microscope and all the reagents and all the tools you need to do microscopy. And then we're gonna let you get settled, and then we're gonna teach you how to identify mushrooms. So the first day we get back, we'll focus on macroscopic features. So those that you can see with the unaided eye, and we'll key out a bunch of mushrooms together, along with me and the TA. We'll guide you through it and teach you how to use the keys, the field guides, right? And then we'll turn you loose, and you're gonna you've been out collecting mushrooms all day, so you're gonna try and identify all the stuff that you collected, and we're there to help you and tell you if you're right, or sort of point you in the right direction if you're not. The second day, we'll teach you how to use the microscope. Okay, we'll teach you how to use all the tools, how to properly make a section of tissue and look at it under the microscope. We'll teach you how to use different stains, and then we'll teach you all the different micro morphological features that you need to identify. Okay, and so I think this is what makes this class really unique and different from other field courses, is that you actually come up to the field station and you learn how to become proficient in using a microscope and using it to identify fungi. And this is not a skill set that many folks have. So when we teach this class, we get a handful of students from the university, but we get a lot of folks that are doing continuing ed for their jobs, or they're hobbyists that want to take their identification skills to the next level, or they're folks that work for different state or federal agencies that want to come and learn how to identify fungi as part of their job. So lots of different reasons to take the course. Everyone walks away with a really strong skill set. And then of course, at the end of the week, we have a final exam. And so what you're looking at in the left-hand picture here, this is one of the students. These are all the questions. So each one of these little white 3x5 cards has a number and a question on it. And that question pertains to the mushroom. So it's just a nice way for everyone to gauge how much they've learned and to test the skills that they've learned during the week. Everybody gets an A, so it's not a big deal. If you come up and you put in the effort, you're gonna do just fine. All right. So I'm not going to go into this stuff in a whole lot of detail, but I do want to show you some pretty pictures from the microscope to get you excited to hopefully come up and take the course with us. And so when you come up, we'll be going over all the macro and all the micro morphological features of mushrooms or mushroom forming fungi. And you're going to learn a lot of terminology. Like any science, mycology has tons and tons of terminology that's been generated over the years. I try and keep that to a minimum and just, teach you what you need to know. So, the basics of what are all the different tissues, the parts of a mushroom, what do we call those? And then if we think about it, all those macroscopic parts of a mushroom, we also look at them under the microscope at the cellular level. And so when we do that, we're zooming in, we're looking at them in a very different way. So of course we have different terminology for the microscopic features as well. And you'll learn all of these. You'll learn these by sitting through lecture in the mornings, but you'll also learn all these terms just from using the field guides to identify the mushrooms you've collected. All right, computer. Again, this is just showing you some of the tissues and the cells and things that you're going to be looking at. And gives you an idea of how much terminology there is that surrounds this field. So what you're looking at here, this is a cross section of the pileus, in other words, the cap of the mushroom and the gill. And kind of hard to orient yourself, but when we think about these images, what you're thinking about is this. So here's your normal mushroom. Imagine you took a razor blade and you just sliced a perfect thin rectangular chunk out of that mushroom and pulled it out. What you'd be left with is the cap shown here in this sort of pale green color. And then all these little finger-like things sticking down, well, those are the lamellae or of the mushroom. Okay, and if we zoom in on those with our microscope, what we see is that those lamellae are composed of tissue and then all the cells that make the spores. So this is really the site of reproduction for this fungus. All those cells are called basidia in this case because it's a basidiomycete, and the basidiospores are being produced on those. So this is where sex happens for this fungus. Okay, the whole point of producing that mushroom is so that that fungus can undergo reproduction. And this is the actual site of reproduction where it's producing its reproductive propagules. And these are what's going to be basically launched off of these little tiny cells. And then these little tiny spores are going to get dispersed by wind out into the environment. So what do they look like? Well, some of them are absolutely amazing under the microscope. So what we see is that spores come in all sorts of different sizes and shapes and ornamentations. So here is just a small sampling of some of the spores that you would see if you came up and took the class. So Entoloma species have these really crazy spores that have these angled walls on them, okay? Some Inocebi species have stellate spores, meaning that they have all these little projections that make them look like a star, hence the stellate term. And then we have also ornamentation. So this is a Lactaria spore down at the bottom here. Sorry, my mouse is hiding everything on me. What we got here, or what we observe in these spores, is that they have this reticulated wall. Basically, it's a reticulum. It's an elevated portion of their cell wall that sticks up like little, like the Great Wall of China running over the surface of the earth. It's like that. And it forms these little fins and little interconnected ridges all over the surface of that spore. In this case, we used a histological stain which stains different sugars, polysaccharides in the cell walls, different colors. So those little ridges that form those reticulations stain intensely blue. So you get this little blue reticulum all over the surface of that spore. And then this is a Gaudiaria spore. This is from like a truffle-like mushroom. And this one has these crazy sort of longitudinal ridges that run, from pole to pole on it. So a huge amount of variation. This is just a small sampling, but spores can be really amazing under the microscope and really fun to look at. All right. We also, as I said, we use histological stains up there, and we'll teach you how to use all these as well. So with spores of fungi, based on the different polysaccharides that are in the cell wall, so whether it's got amylose or dextrose in it, you can get different reactions using this stain we call Melzer's reagent. It's basically an iodine-based stain. That iodine stains the sugars differently. So some things turn bluish in it. We call that an amyloid reaction. Other things turn reddish, and we call that a dextrinoid reaction. And these are things that you'll need to determine to use the actual keys and the microscope to identify fungi to species. And then we have all the other tissues to look at. So the pileapellis, which is the very surface of the cap. So if we go back to our little mushroom drawing here, this is the pileus, also known as the cap, that outermost layer of tissue. So it basically just like your skin, right? On the very surface of that mushroom is referred to as the pileapellis. The structure of that pileapellis can be incredibly variable. So if you think of all your favorite mushrooms, right? We know that some of them are perfectly smooth. Some of them have little tiny hairs all over the surface, and maybe they look velvety. Some are intensely velvety, and they might, even like water droplets get repelled and roll off of them. So there's all that morphological variation that we see at the macroscopic level. Keep in mind that all of that is due to differences at the microscopic level. So this is just another sampling here where this is called a trichodermium, so that the very top surface of that pileus, of that mushroom, has all these little narrow, tiny hairs sticking up. This one is called an epithelium. So much like our own skin, it just has a surface of smooth cells. So the cap of that mushroom would be perfectly smooth. We get one that has broom cells. They're called broom cells because they kind of look like an upside down broom. They're these cells that have all these finger-like appendages sticking up. So you can imagine that a mushroom cap that had those would look like this really dense velvet, okay? And it's really amazing if you look at it with a magnifying glass. And then there's also a cutis. So much like an epithelium, a cutis is just smooth hyphae that's running in a radial fashion. And that kind of gives the surface of the mushroom a smooth, fibrous appearance. Okay, so all of those morphological features that we observe are underlain by microscopic features that we can see with the microscope. And then finally, for the last thing we'll look at here, and this is my all-time favorite, these are cystidia. Cystidia are sterile cells that we find on the mushroom in different locations. And they take on all sorts of crazy shapes. They can be different sizes. They can be pigmented. They can be clear. They can be sharp. They can be dull. They can have thick walls or thin walls, and they can have ornamentations all over their surface. They're totally bizarre. And the really cool thing is that they are the same from one specimen to another of the same species. So they're genetically determined and they're conserved, meaning that if you look at one mushroom, so you look at Mycena navicola, this is our little snowbank Mycena that we get in the Sierra. If you look at the chyla, or you look at the cystidia of one specimen, and then you drive 100 miles away and you collect another one, you look at the cystidia of that specimen, they're nearly identical. So they're conserved, is what we would say evolutionarily, or genetically controlled. We don't know what they do. We have some hypotheses about what they might be doing out there, but I think they're really just there to make mycologists go, what's that there for? So in many cases, we can hypothesize what their function is. We think that their function might be to help keep the cells separated and help maintain the humidity and the moisture around the cells that are going through reproduction. Some of them have these crystals, so they might have calcium oxalate crystals on them. You can imagine little insects that come along and bite into that calcium oxalate. It's not going to taste very good. So that might be a way to deter things from eating the actual mushrooms. Some of them are also very sharp and pointy and thick-walled, so that could also be something that might deter insects and other fungivores with tiny mouth parts from eating them. But they're really amazing under the microscope, and by far, every time the students see them, they just, they can't believe what their eyes are seeing. So we do find them on different parts of the mushroom. We find them where the spores are being produced. So the gills are known as the hymenophore. So we have some that occur there. We have some that occur on the pileus surface in some species, and some that occur on the stipe surface in some species. Okay. And to show what some of these things look like, I've got a handful of photographs here. So these are the sort of real basic, kind of, interesting, boring shaped ones, but, you can have this weird little taper to it. So it looks like a bowling pin, or like a, balloon animal or something. Here's one that really does look like a bowling pin. So just this little pinhead on it, and then this big fat base, like a big fat bowling pin. Here's an image. These are those crystal incrustations. So these are calcium oxalate crystals that are produced on the outer surface right at the tip. So you can imagine when a little arthropod or a little insect came and took a bite of that, it's going to be very, very bitter and sour in their mouth, and that might deter them from eating the mushroom. And then, of course, there's the genus Mycena that I worked on. And Mycena just has the most insane, crazy looking cystidia, basically in most of the species. So this one down here reminds me of balloon animals, right? It looks like you're at the circus. Here's this one that has these big sort of club-shaped cells that are covered entirely with these little tiny cylindrical nubbins. And then at the very tip, it has this one very, very long appendage. And then here's one that just reminds me of a lab, one of those, like if you blew up a latex glove, it looks like that. It's just this big fat cell with all these sort of finger-like appendages sticking off of it. So they're really incredibly wild to look at under the microscope. And what's really great about cystidia is that, as I mentioned, because they're conserved from one individual to another, they're taxonomically really, really informative. So for certain groups of mushrooms, looking at the cystidia will be one of the most important things you can use the microscope for to help you identify that mushroom to genus or to species. And just a few more. These are ones that occur on the stipe. And so this is, we call those caulocystidia. And if you look at these ones, are these long, crazy cells. The entire surface is densely covered with little tiny spines. And then I love this one. This is, the species is Hemimycena tortuosa. And if you look at these, they're actual corkscrews. So this one is great. They're actually a corkscrew cell. It's a long filamentous cell that is shaped like a corkscrew, which is known, it's called tortuous. So that's Hemimycena tortuosa. And then here's some from the pileus of that little Stribularis diminutivus. These are just these sort of bowling pin shaped cells that cover the entire surface. So if you look at that mushroom macroscopically with the hand lens, because it's so small, you'll just see all these little hairs sticking up. And if you put the whole mushroom cap on the microscope and look at it, you see all of these really amazing cystidia just standing straight up, which gives it that texture macroscopically. All right, so that's it for the pretty pictures. I just have a few more slides left where I just want to sort of tell you, what do we know about fungal diversity up in this region of the Sierra Nevada? And so as I mentioned, every time we teach this class, we find a number of taxa that are new to our list. So we've been keeping a list of all the species we find ever since this course was taught for the first time in 1982. And so what we see is that certain years, we don't find anything new for the list. Other years, we find a huge number of species that are new for the list. And what's really amazing is that most of the time we have a big spike. So in this case, we have 19 species that are new. We'd never seen them before is what that means. 19 species were documented up there during this week in 97, and then again in 03. In 97 and 98, those were 2 years where postdocs came out from Harvard and they worked with us. They came out and took the class, and they focused on ascomycetes. And ascomycetes are cup fungi and their relatives. And there's a bunch of them out there that we had just never really taken the time to look at. So they added a bunch of new species to the list. In 2003, that's when I was working on my PhD. I came out and I did the same thing. So I came out and was focusing on a really specific group of very small cup fungi that most people just ignore. So I was able to come out and identify and add a whole bunch of new things to the list. So, some years are really great. And what's interesting is that the more sort of the less productive it is in terms of big fleshy macrofungi, we usually have more things added to the list. So if it's a year where the students are out really looking hard for mushrooms because it's either dry or it's cold, whatever it might be, those are the years where we find new things because the students are bringing in everything rather just than focusing on the big giant, mega fungi, the big charismatic mega fungi and things like that. So we tend to find, new species in the really off sort of dry years. How many have we found so far? We're up to about 380. So if you look down here, this is from 82 to 86. In that period, as far as our records go back, we started out, we're about like 175, 180. And then in the years since then, every year, we're still adding more. And if you'll notice this rarefaction curve, it's not leveling off, meaning that, you know, we're still on a pretty good incline, that there's still a number of species to find and document up there that we haven't found yet, which is really exciting for me. And then how do we do every year? So what's our species count by year? Meaning how many species do we find during that week the course is offered? And this is always, I always make this a competition where I say, you know, we don't want this to be the worst year ever. You guys have to go out and find more than we did last year. So if we look, it's, you know, pretty consistent. So here, you know, under 140, the biggest year we ever had in this, we had about 170 species, I think. And the worst years we've ever had were the last two. But that's not because the mushrooms weren't there. It's because there wasn't a whole class of people out looking for them. So I went up the last 2 years with just a few friends, and you know, we walked around, we checked off as many species as we could find, but we only got about 50 or so. So it tells me that, you know, the students are the ones really bringing in all the material that helps us keep our numbers up. Now, looking at this, you know, you see there's a pretty good amount of variation, and this is going to be dependent on climate. It's going to depend on the amount of snowfall we had the winter before. It's going to depend on how dry or cold the spring was, how much spring rain did we get, how quickly did the snow melt, how warm is it or cold is it during the week the class is taught. There's all these different environmental factors. And a couple of years ago, I was convinced that I could go and I could, you know, correlate this with a bunch of the weather stations that are, you know, found in Truckee and other areas close by. There's really no rhyme or reason. So you imagine that during these incredibly heavy snowfall years, we'd have more mushrooms. Not always the case. You might think that if it was an incredibly wet fall, we'd have more mushrooms. Not always the case. What we do know is that mild temperature and mild weather and rainfall in spring definitely gives us really great mushroom hunting conditions. So as you can imagine, this past spring hasn't been really good in the Sierra. Excuse me. We had really high temperatures up there in early June and late May. And what we saw was that most of the snow, rather than melting into the ground and saturating it, was just sublimating. It was basically just evaporating because the conditions were so hot and dry. So for those of you that were hoping to take the class this past year, you might have been okay. We might have, you know, we would have found a lot of species if there had been more students up there, but it wouldn't have been one of those phenomenal years where there's just boletes all over the place and tons of mushrooms. Okay. All right. And then finally, I just want to say what I've started doing in the last couple of years, just so we can keep tabs on this and also make you folks feel like you're part of science, is we started really incorporating citizen science into this class. So Dan Madrone, a friend of mine down in Nevada City area, San Juan, he and I started a project, and it's a mycoflora project, which has a different name now called Fungus, but we started a spring fungi of the Sierra Nevada project. And so what we're doing is we're trying to really document every single species that occurs in the Sierra Nevada, you know, both in spring and fall. So what I've done is I've created an iNaturalist project that is unique to this class. So all the students that come up can bring their phones with them, and they can, you know, take pictures of all their observations, and they can upload these to iNaturalist. So we actually have a really nice database that's being built that includes, you know, the geotag data for all of these, the data was collected at, the elevation, all that wonderful information is now being put into iNaturalist. So if you get a chance and you want to see what we have found over the last several years up there, go ahead and go, you know, have a look at this. But this is something you can absolutely participate even if you're not in the class. Okay. All right. With that, I will say thank you, number one, to the friends that are surrounded the field campus for all the wonderful things you folks do. The field station is in desperate need of TLC, and you folks are providing it. And I can't wait to someday have a new, shiny, light, warm lab that we can all work in. I'd like to thank Sharon Lane. She was wonderful. She was very interested in fungi. She kind of spent some time with us up in early June out looking for mushrooms. And then, of course, photo credits, not all of those are mine. So I just want to thank the folks I pilfered those photos from. And with that, thanks to all of you for your attention.
Thank you, Brian, for fascinating fungi. So do the students need to have any special equipment to take the class?
Not at all. So we provide the microscopes, we provide everything they need for that. The only thing that students need to bring with them is something to collect with. So whenever someone enrolls in the class, I send them an equipment list that we recommend, and that's a basket. So some sort of sturdy basket that you can collect mushrooms in. We like to use little plastic tackle boxes to put the more delicate, small specimens in, and then something to dig the mushrooms up with. That can be a pocket knife, as you saw in my video. I just use a pocket knife, but you can also bring a garden trowel or something like that. And then I just give them recommendations on clothing, just layers, sturdy boots, bug repellent, sunscreen, rain jacket, snow hat, gloves. So we never know what we're going to get. So it's always
Right.
different.
You want to stop sharing and maybe we can ask
Yeah,
everybody to turn their
absolutely.
cameras on so we can say hi and people can unmute if they want to. Um, so did I tell you that we have a really special donor who has offered to match all the donations that come in for our Zooms this summer? And this is our last one. So she's offered to match donations up to $10,000, which is going to be huge for the lab. So if you haven't donated, you can go to friendsofsnfc.org and your donation will be doubled. I have a message here that you can't turn on video.
Ah,
Okay, there we go. Okay, now you can. So that was a pretty exciting uh, donation.
that's great.
Yeah. And I don't know if anybody has questions about the field campus itself, the logistics of staying there in what can be very cold weather. There's lots of places to stay off campus. As well, you can stay on campus in our tents, and so you have a lot of options. And the food is fantastic, especially if you get lots of boletes, right?
Yes, it's really, really great in
Yeah.
bowl eaters.
So
Wow.
I want to introduce Diane and Bill Wilson. Wave, so everybody can see you. Do you want to say a few words? Bill's the president of Friends. His son is Dr. Wilson, who comes every year, and they come every year. Do you want to say anything? You're muted.
We've been it was the mycology class was the reason that we started living in this area and going up every year. Uh, we have to, the previous director, Jim Steele, made the mistake of hiring our son to bird band for one season, and we got hooked. And he went to the dark side and decided to go into mushrooms, and he's been active ever since. Matter of fact, one of the pictures that Brian showed of the classroom,
Mhm.
they were all in there. Dan Nicholson was in there when I took it 15 years ago, and Andy was in there, and
Even JR
several
was in
other
there, right?
alumni when Brian was the TA for the class. So it's been going, and we've been there for 2-3 snowstorms. Okay, and you live. It's it's a little bit. The nice thing is the dining hall is nice and warm, so you can survive.
And it makes it memorable, right?
Right.
Uh, And one of the Jill, we'll get to your question in a second. Oh, you answered her question. She wanted to
Yep.
identify that mushroom. One of the really awesome things that happens at the field campus is this cross pollination of learning. Because while the fungi week is going on, there's probably the bird song or birds of the Sierra Nevada going on, and you have the opportunity to sit in on talks of the other classes that are happening there. It's just really neat opportunity that goes far beyond what you signed up for. So I put in the chat, and let me just do it again. If you want to copy and paste, I'll give you a minute before we sign off here. The link to donate, if you want to be on our mailing list. That we have a YouTube channel that has lots of videos of the Zooms that we've been doing. I've got the Facebook links. I've got Brian Perry's website and the species list for the spring fungi class. So just copy and paste it into a document and you'll have all that at your fingertips. And people are signing off. So, oh, I should introduce Steve. Steve, say hi. He's also on the board of the Friends. You're muted. I don't know if you've met Brian.
Hi, Brian.
Hey there.
Hello from 5.1 miles from the field campus at my cabin between Sierra City and Bassett. I'm closer to Sierra City. So Google tells me I'm 5.1 miles away. So if you need for me to run up to look at anything up there, I can do it. That's what I'm saying. It's warm here. I don't think I want to tell you how warm it's this weekend. It's supposed to be 99 in Sierra City and 104 or 5 in Downeyville. Yeah. But we're cooling down after that. You know, we're all a little challenged this year in California with the heat and drought, but we're all hanging on and looking forward to more propitious, damp weather for mushrooms in the future. But thank you. I know Jill Horn had asked me, and you responded to her when we posted about this great Zoom, which was terrific. She asked me the name of that specific mushroom, and I said, I'll ask Brian or join us, and I'm so glad she did, and you responded to her, so that's terrific. Because they're beautiful. I mean, I think people were struck by how beautiful and amazing many of these organisms are.
And Jill is a good example of this cross-pollination that happens because Jill is an artist and was in Lori Wiggum's Zoom about a month ago and heard about this one. And hopefully you've been doing some sketches. Yes, she has.
All right.
So thank you. Well, does anybody have anything they want to share or ask or comment on? If not, we'll say goodnight and thank you so much, Dr. Perry.
My pleasure. Good to see everyone.
Yes, we hope to see you next year in a beautiful lab.
It was terrific. Thanks, Brian.
Yeah, that's
My pleasure.
Fantastic.
Always happy to
Fantastic.
Yeah.
Thank you all for all your hard work.
Brian, say
everybody.
hi to Andrew when you see him. Give him a
Will
big
do.
hug for me.
Okay. Yes, I'll see him on Tuesday. That's when I'll be arriving in Denver. Yep.
All right. Bye, everybody.
All right.
Bye-bye.
See y'all.